Hello dear students, welcome to GTC. In this video, we are going to discuss the previous year question of Gpat, that is Gpat 2023 shift 1 question of other subjects. So is this video basically consisting of detailed explanation of each and every questions that asked in Gpat 2023 shift 1 under the other subject category. So let's see what was the first question. The first question was establishing a complex structure of dash is more complex problem than others. So your options are polysaccharide, protein, nucleic acid, peptide. So if you remember when I was talking to you guys with the carbohydrate topic, I have discussed. So polysaccharides are the more complex structure. Why like that? So these are consisting of, or they are monomers of, not exactly monomer, they are basically multiple units of your monosaccharide. Okay, and these monosaccharide joints by these are joined by glycosidic linkage. Okay, they are joined by glycosidic linkage. This linkage may be alpha 1 4, alpha 1 6. There are many type of linkage that has been there. So these monosaccharides are joined together by glycosidic linkage and to form a polysaccharide. And these polysaccharide, they are either they are linear or they are might be what we can call branched. Okay, either they are linear or they are branched. That's why these polysaccharide having a complex structure as comparison to protein, nucleoside and peptic acid. Let's see the explanation. Polysaccharide are simply called as glycan. They are consisting of a repeated unit of monosaccharide, what we have discussed, that is multiple unit of monosaccharide, and they are basically joined together by this glycosidic linkage. It may be alpha 1, alpha 1 4 1 6 1 3, or beta 1 4 1 6 1 3 type of linkage. Polysaccharides, either they are linear, that means straight chain, or they are branched, which you can see these are the branch or more complex structure. However, in contrast, the structure of protein and nucleic acid are only linear polymer. Please remember, protein and nucleic acid structure are more linear. Proteins are basically higher molecular weight mixed with a polymer of alpha amino acid, and they are joined by peptide linkage. Glycosides are joined by polysaccharide are joined by glycosidic linkage. Proteins are joined by peptide linkage. Okay, so this peptide structure you will see. So question can be asked that how this peptide structure is formed. So please remember, the peptide structure is formed by the fusion of this carboxylic group and your amino group. This is also one important question. Question can be asked. The formation of peptide bond in a protein is due to fusion of your carboxylic group and amino group. Okay, so the reference has been taken from your biochemistry book by U. Satyanarayan and U. Chakrapani, fourth edition, page number 2024-53. Also, these things are present in your GPAT mantra book, which is uh, available in our GDC classes app. You can purchase that book. Next question. Which one is the most characteristics of hexose monophosphate pathway? So HMP pathway. So let's recall what is HMP pathway. So if you remember, I have explained you that HMP pathway, this is basically a alternative. Okay, this is a alternative pathway to glycolysis and TCA cycle. This is a alternative pathway to glycolysis and TCA cycle. Okay, alternative pathway to glycolysis and TCA cycle for oxidation of glucose. For oxidation of glucose. So few things I want to remind you that this is basically this HMP pathway. This is an alternative pathway for glycolysis and TCA cycle, and this is basically start from which compound? Glucose 6 phosphate. The starting material is glucose 6 phosphate. One thing that always asked in exam, and this question also based on this same thing, that in HMP sound or HMP pathway, no direct production or utilization of your ATP. Okay, no direct production or utilization of ATP. Let's see. Question is which is not the characteristics. First thing, it produce carbon dioxide. Yes, carbon dioxide is produced. It required ATP for phosphorylation. No. Just now I have mentioned in the point, no direct production or utilization of ATP. It controls the inhibition of glucose 6-phosphate dehydrogenase by NADPH enzyme. It produces ribose 5-phosphate. Yes, it is also correct. So the correct answer is option number B. Okay, so this is an alternative pathway to glycolysis and TCA cycle for the oxidation of glucose. The pathway basically starts from the intermediate glucose 6-phosphate. Here, no ATP is directly utilized or produced in this HMP pathway. This is having two pathway, that is one is your oxidative pathway, another one is your non-oxidative pathway. One is your oxidative pathway, another one is your non-oxidative pathway. For the complete oxidation of glucose 6-phosphate, 6 carbon dioxide have to be to 6 carbon dioxide, we have to start with 6 molecule of glucose 6-phosphate. The overall reaction is 6 glucose 6-phosphate, 12 NADP, 6 H2O. It will produce 6 carbon dioxide, 12 NADPH, 12 hydrogen, and 5 glucose 6-phosphate. Okay, so this is the cycle. This is basically start from this glucose 6-phosphate. This is also called as pentose phosphate pathway or phosphogluconate pathway. Okay, starting from your glucose 6-phosphate. Here the intermediate is ribose 5-phosphate is one of the intermediate. Okay, so the reference has been taken from the standard book of biochemistry by U Satyanarayan and Chakrapani. Okay, so and the all this MCQ detailed explanation are also present in our Gpat mantra book. This Gpat mantra book contain the detailed MCQs with the detailed explanation of each the previous year questions from 1988 to 2024. Okay, this is the highly recommended book for the preparation in last minute and to boost up your preparation, because basically if you will analyze the previous year question, you will have a greater clarity that what are the pattern of the question, what are the trend of question that has been asked in the exam, and how to prepare, which part of the particular topic has to be taken care more prioritizedly as compared to others. So these things or these analysis basically has to be what we call has to be analyzed with this GPAT mantra book. Okay, so moving to the next question. List one, match the list one and list two. I have, whenever I discuss this type of question, I always explain you, in this type of question you have tried to find out one answer. If you have, in 90% cases, if you know one correct answer, then the rest of the answer will be automatically correct. So we have this name of the vitamin in list one, and second list we have function of that particular vitamin. So riboflavin, vitamin B1, B2, thiamine, and pantothenic acid. Okay. So this is your riboflavin, thiamine, vitamin B1, riboflavin, vitamin B2, niacin, vitamin B3, pantothenic acid, vitamin B5. Let's match. So we have five options. So you know vitamin B5 is always act as a coenzyme. Please remember, this is one of the important question or important one liner also. So this question can ask in NIPER also. So pantothenic acid is act as a coenzyme. So four option is by default will be R. Next we have remaining thiamine, niacin and riboflavin. Okay. So please remember what? Niacin. Niacin having the coenzyme is NAD and NADP. Niacin having coenzyme NAD and NADP. And this NAD and NADP basically actively participate in your ETC, electron transport chain, as a electron carrier. Okay, it is act as a electron acceptor. Okay, so option 2 will be, bit niacin function will be in ETC. Next is we have left riboflavin and thiamine. Okay, riboflavin and thiamine. Thiamine or vitamin B1 having actively participate in case of alpha decarboxylation reaction. Okay, so it actively participate in decarboxylation of reaction. Next we have remaining is uh, riboflavin. So riboflavin, this is basically act as a cofactor for succinate dehydrogenase. Okay, it is act as a cofactor for succinate dehydrogenase. So option A is the correct answer. So you have to remember this vitamin and its function. So there are four B complex vitamins are essentially participate in the Krebs cycle. These four vitamin, please remember these four vitamin are actively participate in your Krebs cycle. Okay, this also I have discussed with you in the our theory class. Okay, so let's see the function riboflavin, which having coenzyme FAD and FADP, FADS. So this is function as a coenzyme for succinate dehydrogenase enzyme. Next is niacin, vitamin B3. The coenzyme is NAD and it is involved in isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, and malate dehydrogenase. This is basically causes decarboxylation reaction. This is basically participating in decarboxylation reaction. Also, this NAD and NADP also participate in your electron transport chain. Also participate in electron transport chain. Next is your thiamine, vitamin B1, the coenzyme is TPP. So this is also participate with the act as a coenzyme for enzyme alpha ketoglutarate dehydrogenase or branch chain alpha keto acid dehydrogenase enzyme. This enzyme also requires thiamine pyrophosphate. Okay, next is pantothenic acid. Please remember, pantothenic acid is basically considered the coenzyme is coenzyme A. Okay, so this attached to the active carboxylic residue such as acetyl coenzyme A or succinyl coenzyme A. Okay, reference has been taken from this biochemistry Satyanarayan book, page number 135 to 56. You can check the reference. Next question. DNA and RNA contain the following two major purine bases. So to remember this, there is a simple things you have to remember. We have DNA and we have RNA. Okay. So there are different type of purine and pyrimidine present in DNA. So DNA basically contains ATGC, adenine, thymine, guanine, and cytosine, ATGC. In RNA, it is present is AGCU, AGCU. In place of thiamine, uracil is present. Okay. Similarly, adenine, guanine, cytosine, cytosine, and uracil. So one I ate. So the DNA RNA contain the following two major purine base that is common. Okay, common purine. So common purine is your here purine is adenine guanine. Here purine is adenine guanine. Okay, so option B will be the correct answer. So purine is so we have two type of nitrogenous bases. Two type of Let me explain you in a blank slide. So we have two type of nitrogenous bases. One is your purine, another one is your pyrimidine. So purine. So what are the purine example? Your adenine and your guanine or guanine. Pyrimidine based example is your thiamine, cytosine, and uracil. In case of DNA, thiamine is present. In case of RNA, instead of thiamine, uracil is present. Okay, option B is the correct answer. So it has been clearly mentioned over here. So these are nucleic acids are basically polymer of nucleotides. Okay, they are held together by 3 prime 5 prime phosphate bridge. So nucleotides are consisting of base, sugar and phosphates. You can see the structure of the purine, adenine structure. Okay, 6 amino purine, guanine is 2 amino 6 oxypurine, pyrimidine thiamine name is 2 4 dioxy 5 methyl pyrimidine, 2 oxy 4 amino pyrimidine, next 2 4 dioxy pyrimidine. Please remember this name also important from exam point of view, question can be asked. Okay, so moving to the next question. Match the reproduction and genetic exchange under column 1 with the explanation under column 2. Okay, so we have binary fission, transformation, transduction and conjugation. So transduction means transfer of genetic material with the help of a bacteriophage or virus, bacteria through a virus. So trans option 3 is R. Next is conjugation, the transfer of gene by formation of a cytoplasmic bridge. Transfer genetic material donor recipient through a cell contact, common vegetative reproduction, horizontal gene transfer taking of foreign genetic material. Okay, so conjugation 3, option 3 is R. Okay, option D will be correct. So let's match the other one. Binary fission is, this is the common vegetative reproduction. Okay, transformation is, that is the transfer of genetic by taking up the foreign DNA as a naked DNA, and conjugation is your transfer of genetic material to the donor by recipient through a cell contact, which is called as cytoplasmic bridge. This is called as cytoplasmic bridge. Okay, with the help of a cytoplasmic bridge, it will transfer. So option D is the correct answer. Okay, transformation, that is the transfer of, and this is a method for introducing foreign DNA into the bacterial cells, and the transformation frequency will refers to the fraction of cell population that can be transferred. And this is basically a horizontal gene transfer by taking of the foreign naked DNA. Conjugation, the transfer of this genetic material by formation of a cytoplasmic bridge. Transduction, transfer through introduction into the host cell with the help of a virus, exactly bacteriophage. Okay, next is transcription. Transcription means that is the process which RNA is synthesized from DNA. Binary fission, this is the organism basically duplicates its genetic material. Okay, that is called as your binary fission. Next is transfection and integration. Transfection means, transfection is a biotechnology technique that introduced foreign DNA into a eukaryotic cells to study the function of the gene and its protein expression. This is called as transfection. And the Europe in integration is the European fraction of biotechnology, broadly considered biotechnology as a integration of natural science and organism cells as a parts of thereof or molecular and analog for the product and services. Okay, you can take the reference from this author U Satyanarayan and Chakrapani book, first edition, page number 39, 87 to 89. Okay, as well as from the pharmaceutical microbiology by N.K. Jain, third edition, page number 96. Next question, which again match the list of 1 with list of 2. Okay, so here you have to match the fermentation product and their strain use. Dextran, bacterial amylase, glutamic acid and vitamin B12. Okay, let's match. So vitamin B12 is, I know vitamin B12 is basically, for the production of vitamin B12, the strain used is your Streptomyces olivaceus. Okay, next is glutamic acid is produced by the species of Bacterium species. Okay, next is bacterial amylase is basically produced by the bacteria your Bacillus subtilis. And what is left? Left is your dextran. So dextran production is basically carried out by leuconstack mesenteroids. Okay, so option D is the correct answer. This has been already been discussed several times, this table, in our theory class, and I have repeatedly told that this topic is important, this topic is important, this topic is important. All you have to read this chart which I have discussed in our theory class. So very carefully you have to remember all these things. So let's see the explanation. Yeah. Dextran. So dextran production is basically carried out by leuconstack mesenteroids, Acetobacter species, and Streptococcus mutans. Next is your bacterial amylase. So bacterial amylase production is basically carried out by Aspergillus oryzae, Aspergillus niger, Bacillus subtilis, and Bacillus laciniformis. Next, glutamic acid production is carried out by Mycobacterium species, Brevibacterium species, and Arthobacterium species. And last one is your vitamin B12 production is taken care by Bacillus mesenterium, Streptococcus ovirus, Butyribacterium retrograde, Microspora species, and Propionibacterium petronici. Please remember this name. This is very, very important from exam point of view. Moving to the next question. Given below are the two statements. One is labeled as assertion and one is labeled as region. So you have to match. Okay. What you have to do? You have to match. So assertion is exotoxins diffuses freely through the bacterial cell wall into the medium in which organisms are growing. Yes, correct. They are water soluble and can pass into the surrounding medium. Yes, both are correct. If you remember, I have explained you details. So option A is the correct answer. So I will explain you. So whenever I was talking to you, depending upon the function of exotoxin and endotoxin, we have discussed very carefully about their explanation. So exotoxins are basically produced by both gram positive and gram negative bacteria, but majorly by gram positive. Exotoxins are basically heat labile protein that can easily degraded by heat, and they are either actively secreted from the bacterium or released upon bacterialysis. They are basically water soluble and can pass into the surrounding medium. Okay, they are basically water soluble and easily pass into the surrounding medium. Exotoxins significantly play a role in virulence factor for causing disease. Once after the toxins are liberated, it diffuses into the surrounding medium and causes harm to the host of the cell. Okay, so this information was taken from the book Pharmaceutical Microbiology by Astoskar, page number 321. This is the detailed explanation and colored explanation is there in our Gpat Mantra book, one of the most highly recommended book for your preparation to boost up your preparation of Gpat Hyper Journey. Okay, so moving to the next question, basic dye. Okay, basic dye that are used in microbiological staining, which of the following statement is wrong about the mechanism of staining? So basic dye. Okay. The positive ion on the surface of the bacteria form a color complex with dye. Ionic exchange between the negative charge of the bacteria and positive charge of the dyes takes place. Ionic exchange between the positive charge on the bacteria and the negative charge on the dye takes place. The neutral charge on the surface of the bacteria forms a color complex with the dye. Which of the following statement is wrong about the mechanism? So the correct answer is option D. Let's see the detailed explanation. So mechanism of staining that involves the ion exchange reaction between stain and the component to be stained. So based upon the nature of the chromogen, they are of three types. Either they are acidic or anionic dye, basic or cationic dye, or neutral dye. First is acidic dye or anionic dye. Acidic dye, this is basically colored the negatively charged and hence called as anionic dye. It usually stains it used as a stain basic part. Basically acidic dye basically used for the staining of the basic part of the cell. Okay, such as your histone protein and background staining. Since most of the bacteria cells are negatively charged, the acidic dye does not stain them. This is because the repulsing the charge. Because there is interaction between positive and negative. There will be never attraction between negative. So between the same charge, between the opposite charge, there will be attraction. So please remember this principle between opposite charge, attraction Okay, between same charge, between same charge is repulsion. So that's why repulsion is there. So a acidic dye example is your eosin, nigrosin, Indian ink. They are the acidic dye. So similarly, if we'll talk about basic dye basically called stain the positive charge. That's are called a cationic dye. They usually stains the acidic part of the cell, such as your bacterial cell wall. Example is your methylene blue, crystal violet, sulfurin, basic fusin, and malachite green. Neutral dye, they stain both your acidic and basic part of the cells. Example is your Gimsa stain. So this is the detailed explanation you can see from our Gpat mantra book. Moving to the next question, which of the following represents a complete list of product that are offered by company for sale? Product variety, product mix, product item, या product line. The correct answer is your product mix. So what is these things? A product mix, this is concerned with the combination of the product offered by the firm for sale. If we talk about what is product variety, this is the range of different product as a company may offer within a specific category or product line. Product item refers to the specific version of variant of product with the production line. Production lines refer to related to the product that are marketed or under a single brand. Okay. So reference you have taken from Pharmaceutical Production and Management by CBA Subramaniam, first edition, page number 199. Okay. So moving to the question number 124, shift from try my product to prefer my brand marketing strategy is done at what stage of product life cycle? Introduction, growth, maturity, decline. That means you are initially when we see. I will give you a simple fundamental example. You will understood this company. So for suppose you have made a startup for making of jeans. Okay, or making simple example. We as a pharma students, we let's take an example of a product. So we let's you have a developed one inhaler of XY drugs. Okay. So when you first develop that drug, you will give this product to different age group or different physician or different patients to try their product. Okay. Once this product get what we call once this product get uh, market popular under the among the people, now this product will going to be your brand. Okay, that is basically going to be your brand for uh, your selling and other purposes. That's why, so when your product get popular, you will approach towards the growth. So that's why in growth phase, okay, in growth phase, basically the try primary product to prefer my brand, basically which changes occurs. So option B is the correct answer. Okay, so growth basically when increase in sales increases, we take on the transfer to try my product to prefer my brand. Okay, they convert try my product to prefer my brand, and this is basically strategy and that has been strategy for the product life cycle. Moving to the next question, hemophilus influenza type B vaccine is an example of subunit vaccine, toxoid vaccine, recombinant protein vaccine, or conjugated vaccine. So hemophilus influenza B is basically a conjugate vaccine. Okay, what is exactly conjugate vaccine is? So Haemophilus influenza type B is an example of third generation polysaccharide conjugated vaccine. This is an example of third generation polysaccharide conjugated vaccines that have recently been introduced by many national immunization programs. Okay, so these vaccines are comes under a conjugated vaccines, and this is an other example of conjugated vaccine includes your pneumococcal vaccine and your meningococcal vaccines. Okay, so these has been all the detailed explanation has the present in our GPAT mantra book. Yeah, this is highly recommended for this to boost up your preparation. Okay, so thank you so much. In the next video we are going to discuss what are the questions that has been asked in the GPAT 2003 shift 2 exam. Okay. Thank you. Thank you so much for attending the class.